Good morning. My name is Prarthana Dalal, and I'm from Kansas City, Kansas. Currently, I'm a freshman at Northwestern University in Evanston, Illinois, and I'm a pre-medicine biology major. The research that I want to present is on understanding how sickle cell disease, how we can understand the hemoglobin genetics behind sickle cell disease. So the idea behind my project was to understand how a small change in the promoter region of a gene called gamma-globin can help increase its expression or production in the adult stage, so it can serve as a replacement for the defective adult beta-hemoglobin gene in adults that have sickle cell disease. And for this, I designed two types of mice models. These mice had the mutation that I was looking at, this -175 T to C and the -195 C to G changes, and were transgenic, so these had a construct of the human genetics in these mouse models. Eventually, what I found was that both these mutations upregulated gamma-globin production, which is a promising result for future. This shows us that we can look at pharmaceutics and target this region of the genome so that in the future we can upregulate gamma-globin to reduce the severity of symptoms in patients with sickle cell. So sickle cell disease is caused by a mutation in the beta-globin gene that translates over to an amino acid change. This other type of hemoglobin, called hemoglobin S or sickle hemoglobin, doesn't have the same efficient oxygen carrying capacity as normal hemoglobin does. As a fetus, people have a different form of hemoglobin that's produced that has a higher oxygen affinity so that oxygen can be extracted from the mother's placenta. This production of this gamma-globin or fetal hemoglobin stops as soon as you're born, and a switch occurs to the production of adult hemoglobin. The goal of my project was to try to understand how we can change the sequence of this fetal hemoglobin so it can continue being expressed in the adult stage, so it can replace that defective adult hemoglobin. And for that, I designed a construct called the beta-yeast artificial chromosome. This had the entire sequence of both the fetal hemoglobin gene and the adult hemoglobin gene, and it was put into a mouse model, so these mice express these human genes. This was a very unique aspect of my project, and understanding the cloning and the molecular genetics behind that can also be applied to a variety of other different biomedical research fields. And so ultimately what I found was the methodology behind that, and also understanding that these mutations, these one sequence changes, can increase expression. This leads us to perhaps hypothesize that there are specific proteins that bind in this region that we want to be able to identify in the future that can be targets for therapeutics.